Okay, our next speaker is Dr. Barbara Bailus, who is actually the chair of the FAST Scientific Advisory Board. Just so you all know, she does deserve a round of applause. She's a volunteer and does practically as much work as I do for FAST. She is an incredible asset to our community and doesn't get enough credit. So we should certainly give her a round of applause. Barb is going to be talking to us about designing the ultimate backstage pass to the brain. She's currently an assistant professor of genetics at the Keck Graduate Institute, and she trained with Dave Siegel when he was part of the FAST Fire team, and she was responsible for much of the work of the artificial transcription factors in Angelman syndrome. Barb.
Thank you so much for the kind introduction, Allison. So it's a pleasure to be here with all of you today. I'm actually really excited about this talk. And as Allison mentioned, it's designing backstage passes to the brain. So it focuses on delivery. Main disclosures, I'm basically the co-inventor of this with Dr. Lisa Ellerby. It's a patented delivery system that we call zip delivery. And it was invented at the Buck Institute, where I did my postdoc work. So a little history, although you already got a preview. I was indeed Dave Siegel's graduate student. I'm not going to tell you how many years ago that was, for both our sake. Then I went and did my postdoc at the Ellerby lab. I worked on neurodegenerative disorders there, and that's where zip delivery was invented. I then went and started my own laboratory at the Keck Graduate Institute. It's one of the Claremont colleges. And this is where I picked up Angelman research again, to apply zip delivery for Angelman research. So when I was at the Buck Institute, a big question was, how are we going to deliver CRISPR to the brain? Because Huntington's disease will also benefit from CRISPR, just in a different way. And so I want to take a moment and thank the people in that lab as well that helped with this work. And the idea that we had, because of the work that I did in Dave Siegel's lab, where we use cell-penetrating peptides, was let's use cell-penetrating peptides. But instead of using what's already out there, let's make a new, novel cell-penetrating peptide. And so you can think of this cell-penetrating peptide as the backstage pass to the brain. And this is what we call zip delivery. Now, when we think of the brain, we usually think of the brain having the blood-brain barrier, and we think of it as a wall. It's not actually a wall, so it doesn't really look like this. Instead, it's an intricate network of blood vessels that both surround and infiltrate the brain. But you can think of those blood vessels where they interact with the neurons as having like a gate or a gatekeeper. And to get through that, you need a backstage pass. And so that's what I design in my lab. And we design that so that you can go from the intersection of the blood vessels into the neurons, because that's where we need to get these therapies, right? So how did we do this? We actually took a tiny piece of the Zika virus. So you're not going to get Zika from this. We just took a tiny, tiny piece. And this is what our backstage pass is. Now, when I was a postdoc, we glued this onto CRISPR, but it was a CRISPR nickase. This was great for Huntington's disease. It's not as applicable for Angelman syndrome. So what I'm going to tell you is what I'm now repurposing for Angelman syndrome. But I'm going to show you some of the work and why we think this is going to be promising for Angelman syndrome. So how's it going to work? Well, there's that intersection there between the neuron and the blood vessels. And that's what we need to get our therapies through. So how we do this is we would usually try to send something in, and this is exactly what would happen. Not going through. We put the backstage pass on, and all of a sudden, it gets permission to go through. And you can get into the neurons then and have the therapy that you need. Now, what was really cool about this was for delivery, we actually just inject in the mouse tail vein. Now, I know humans do not have tail veins for obvious reasons. What that would be for a human is you'd basically just be injecting in a blood vessel. And it gets through the blood-brain barrier. So we injected in the mouse, and about 24 hours later, we were able to harvest the mouse brains and see that we had CRISPR throughout the entire brain. And so I think you can appreciate here that although the untreated mouse has some red, which is background, the ZIP CRISPR-treated mouse, which has that backstage pass, has a lot of red signal. And what we're staining there for is actually the CRISPR. So you can see a lot of CRISPR inside this mouse brain. So there's the potential to deliver CRISPR, but because this is a peptide, this little backstage pass, it's mobile. I can give it to other proteins. It doesn't have to be CRISPR. Now, when we zoom in here, this is the hippocampus. This is the region that's really important for learning and memory. I don't need to tell this audience how important that is for your children. But we can get into the hippocampus. That is deep inside the brain. And this is after only a couple IV tail injections. So how am I applying this for FAST? Well, we have two pillars that I'm able to address with this technology. I'm addressing pillar 1, which is replacing mom's gene. Because remember, I said I could give this backstage pass to any protein. So I can give it to UBE3A. I can also hand the backstage pass off to CRISPR, and it can go in and stop the stop. Or it can activate genes or repress genes. So where are we as progress on this? So I've attached the ZIP to UBE3A, and I've put it onto neuronal cells. And seen that, yes, it gets in. That green is UBE3A in the cells. And the bottom one, you can think of as a selfie. It's basically what we call a Western blot, but it allows us to see that the protein is indeed inside those cells. All right? And it's exciting because it's in there in as soon as 4 hours, and it persists for 24 hours. Now, this would be a repeated type of treatment, but repeated as in we're going to do just a vein injection. So it would be like an insulin treatment. And it could even maybe be one day hooked up to a pump. Now, because one cell-penetrating peptide is never enough, we are also working on other cell-penetrating peptides in my lab. So more backstage passes, because not every backstage pass will work for every single protein. And so with that, we have different backstage passes that I've designed. And one of them right now, we've actually attached to CRISPR. And this is a CRISPR activator. And we've put it into the neural stem cells. And we see here that I do indeed both get it in, and we have, once again, that selfie picture. So where are we going from here? Hopefully next year, when I stand in front of you, I'll get to tell you the results of these planned experiments. But I am planning to be testing the ZIP UBE3A in mice. So we'll be injecting the UBE3A with the backstage pass into mice. Then we'll be testing ZIP CRISPR for Angelman syndrome in mice. And we're going to characterize and test those new backstage passes, hopefully both in cells and then in mice again. And hopefully, next year, I have that pretty red picture to go with this so that it's not just a cartoon, but real, actual data. And I'd like to take a moment now and thank all the people that made this possible. So a special thank you to the lab where my Angelman syndrome journey began, Dave Siegel. A special thanks to Allison Brent, who got me back into the Angelman field after I did my postdoc in Huntington's. A thank you to all of you, because you are the ones funding this work, because I have a FAST New Investigator grant. So this is what enables me to take this and adapt it for Angelman syndrome. A thank you to the Ellerby lab. This work wouldn't have happened if that was not the lab that I did my postdoc in, especially to Dr. Lisa Ellerby. And a thank you to my current lab and members of the lab that have now graduated and gone on to med school, who have worked very hard to produce the data that you saw here today. Thank you so much, and if we have time for questions, I'm happy to take them. If not, please find me at some point during today and ask questions.
Yeah. We have time for a couple questions. Can we get us past to Taylor Swift?
Is that for you or your
Because
daughters,
my 11-year-olds
Allison?
would love you forever.
So I have a question as far as this peptide. So is it, since it's crossing the blood-brain barrier, does it have an affinity for the CNS, or do we have to think about having additional UBE3A in the periphery and how that would have an effect?
Yeah, no, that's a really great question. I will say that we do see this show up in the kidneys and in the liver, but whether it's showing up because it's being processed out of the body or whether that's where it localizes to begin with, I don't have the answer. But one of the reasons we ripped this peptide off is because it was shown that Zika does infect the central nervous system. There's a question over here.
Hi, could you explain why this would need repetitive treatments and not a once and done despite the CRISPR technology?
Yeah, no, that's a great question as well. So it's not necessarily that every single use of this would need repeated. It depends on how you're using it. So if we attach it to UBE3A, UBE3A is a protein like any other, so it will degrade in the body. But if we were to attach it to something like a CRISPR nuclease or a CRISPR nickase, where it makes that genetic modification, it could potentially be a one and done. If we attach it to something like a CRISPR activator that Dr. Atov just talked about, those also eventually degrade, so it would likely need to be administered periodically, but maybe not as routinely as the UBE3A.